My name is Terry Sanger. I'm the Chief Scientific Officer at Children's Hospital of Orange County. I'm also a child neurologist, and I work in movement disorders. And a big part of movement disorders is deep brain stimulation, which is a neurosurgical procedure that has the ability to bring tremendous benefit to children with cerebral palsy and other types of movement disorders. And I am very pleased today to have two of my colleagues from the neurosurgery department at CHOC here today, who are going to talk about specifically different neurosurgical technologies and opportunities for intervention in neurosurgery. We have Dr. Michael Muhonen and Dr. Suresh Magge. Suresh is the chair of the department. Dr. Muhonen is co-director of our Neuroscience Institute, and they will be able to talk about several of these technologies. And I'm hoping that you will find this quite inspiring, as I do. So, Dr. Magge, why don't you get started and show us the challenges and innovations that you're working with.
Great, thanks, Terry. I really appreciate that, and really, it's an honor to be here today. So, I was going to start it off with some of the benefits of innovation that we see in neurosurgery, and in particular, how it applies to brain tumors. So let me just share my screen. Okay. So, when we talk about brain tumors, it is a pretty large topic. Pediatric brain tumors are a different breed compared to adult brain tumors. Pediatric brain tumors more often are lower grade, so they can be treated, many of them can be treated with surgery. Many of them are curable, not all. Many of them can be treated with chemotherapy and radiation, and a lot of our kids can lead very good lives after this, after a diagnosis of a brain tumor. And a lot of this has to do with innovation, research, applying the technologies that we have to both surgery and postoperative care. So I'm just going to give a few examples of how innovation has affected brain tumor therapy. And where we need to go in the future, because we still have a long ways to go in particular types of tumors. So one example is this. This is a craniopharyngioma. A craniopharyngioma is a type of brain tumor that affects the base of the brain in the area of the pituitary gland. So the pituitary gland is here. So this is what's called a sagittal MRI, and you can see basically a side view of the brain. The nose is here, the back of the head is here. Above the pituitary gland, you can have this type of tumor form, which has both solid and big cysts around. It's in a very deep part of the brain, and there are different ways that we can get to it. This tumor is technically a benign tumor. So if you can get the whole tumor out, you can potentially cure this patient. But it can be challenging because it is in very deep, important areas of the brain. So this is a picture of what we do in surgery. So this is an open surgical technique where we open up the scalp, open up a piece of the bone, and this is the dura, which is a covering of the brain. And we can see this is the frontal lobe and the temporal lobe. And what we need to do is we have to find some of the natural divides of the brain, separate them out, and then get to the tumor. So if you can imagine, we don't want to necessarily go through normal brain. So what we do is we slide under the normal frontal lobe to get back here. And how do we do that? Well, there are different techniques that we can use. We call it microsurgery, where we go under a microscope, and the microscope is put into the field. We use retractors to retract parts of the brain. This is the frontal lobe, and this is the temporal lobe. And now we're looking straight back at the optic nerves, and the tumor lies just under the optic nerve and the optic chiasm. So you have a right and left optic nerve. This is a carotid artery coming up and splitting. And what we have to do is work basically in between these very thin, narrow areas. So in between the anterior and middle cerebral arteries, just to the side of the carotid artery, or in between the carotid and the optic nerve. And so we go in these small little angles, whichever we can get through at that time. And here I was going just to the side of the carotid artery, coming back here into this space to get the tumor out. And once we take out that tumor, we can see all the way back to the brain stem, which is all the way back to here. So now we're looking all the way back. We take out the tumor, take out the cyst. And then, so this is what it looked like before surgery. And then after surgery, you can see how the tumor is gone. And how do we do this? Well, we can employ technologies that we didn't have in the past. One of them is what's called stealth navigation. There are different companies that make basically the technology so that we can pinpoint exactly where we are in the brain with relation to the tumor. And there are multiple companies, there's Medtronic, there's BrainLab, that make these machines that basically localize different parts of the brain. And so we can use it in brain tumor surgery to get to very specific areas without harming normal areas of the brain. Another technology that we use is an intraoperative microscope. So it's a very powerful microscope that we can use in surgery to magnify very small areas of the brain and use it in surgery. And then the other thing is an endoscope. An endoscope is a camera at the end of this long tube, and we can use it in different types of surgery. In this surgery, I use an open technique, but we can also use, in particular types of tumors, an endoscopic or endonasal technique, where instead of doing a big open surgery, we can actually go in through the nose and go in through what's called the sphenoid sinus and get to the tumor this way without making a bigger incision. And so in particular cases, we can do that. So this is all thanks to innovation and technology that we're incorporating into surgery. Here's another example of a brain tumor. This is called a medulloblastoma. This is one of the most common types of tumors that we see in kids. This is a very large tumor. This whole area is tumor, and it's pressing on the normal cerebellum. This is a child who was 13 months old at the time of diagnosis. We took the child to surgery, and you can see we've taken out the whole tumor, and what's left is just the cerebellum, and it's opened up the pathways. Medulloblastoma, like I said, is one of the more common types of pediatric tumors. And this is a story that's, that shows the change change in the way we treat tumors. With medulloblastoma, the traditional treatment is surgery and then chemotherapy or radiation. About 30 years ago, all these patients got high-dose craniospinal radiation. And we see a lot of these patients today. Some of them may have been cured of their disease, but the high-dose radiation has left a lot of these patients with severe cognitive deficits and hormonal deficits and bad side effects. So since then, the treatments have changed. After surgery, we use different types of chemotherapy. The other thing is, in the past 10 years, researchers have found that these tumors differ significantly based on their genetic makeup, because we found that some patients with medulloblastomas do much better than others. And why is this the case? So a group led by Michael Taylor in Toronto looked at this and found that these tumors actually have multiple genetic subtypes, and some of the subtypes do better than others. And based on these subtypes, we can target them genetically with different types of treatments. So this is some of the progress we've made in the laboratory as a field in looking at how can we treat these tumors better, and how can these patients do better and have better qualities of life after surgery and treatment. I want to talk about a different type of tumor, a non-resectable tumor called a diffuse intrinsic pontine glioma, or DIPG. A DIPG is a very different type of brain tumor. It's a tumor that grows in the brainstem of a patient. So this is the normal brain, the cerebrum. It comes into the brain stem, which turns into the spinal cord. In the back here is the cerebellum. The brain stem is basically what funnels all of your critical neurons down to your spinal cord. So if you have a tumor that lives in the brain stem, many of these tumors cannot be resected because you'd be taking out the whole brain stem. And with a diffuse intrinsic pontine glioma, what it does is it doesn't just grow and push the brainstem aside, it actually grows and takes over the brainstem like this. So this area should be the brainstem, but instead it's taken over by the tumor. For years, the prognosis of children with this tumor never changed. Generally, after diagnosis, the prognosis, life expectancy of a child with this tumor is about 8 to 12 months. After diagnosis, these children would generally get radiation treatment, which might help the symptoms for a while, but it invariably would come back. I would say that this is one of the toughest diagnoses that we have to give to families because of the lack of good treatment options. And for years, we really hadn't made any advances in the study of this tumor. And the reason is that we weren't doing biopsies. And the rationale for that was that biopsies in the past were too dangerous. Because if we took a piece of this tumor in the brainstem, we would leave these children with significant side effects. And we didn't have treatments to give, so we thought there was no point in doing a biopsy if it was not safe enough and there was no treatment to give. But also in that, without getting any tissue, we weren't making any strides in the lab to understand these tumors to eventually make better treatments. That has changed in the past 10 years. So with technology, we have been able to improve the ways and methods that we can to biopsy these tumors. We can use a stereotactically guided needle to go through the cerebellum into the back of the brainstem to take small pieces. And we and other groups have shown that we can do this safely with minimal morbidity to the children. And so I was part of a larger clinical trial looking at this, and we found that we can do this safely with a low side effect profile. Now, what do we gain from this? Well, we can get tissue, but also with our current technologies, we can genetically sequence these tumors to find if there are certain mutations that are particularly amenable to certain treatments. And we're finding new mutations that are in these tumors. And we like to say tumors in children and like this are not, children are not just small adults. These tumors are very different from adult brain tumors. And so now we're learning more and more about pediatric brain tumors, and they can be very different from adult brain tumors. This is an example of how we do the biopsy. When doing this biopsy, we position the child on their side. These little donuts are stickers that we use to basically guide our guidance system. And the system registers these fiducials in space, and then it knows, and it can then correlate that with an MRI. And then in surgery, this is just a guidance wand which recognizes the optical, basically it's an optical tracking system. And then we can put a needle down through this area, and we use this little targeting system to get to where we need to go. And we plot out our path on the guidance system to biopsy a particular area. Then after surgery, it left with a small little incision that gets stitched up. So, this type of work has led to more advances in the field. Here are a few papers that we put out in terms of one. This was a paper we did with a consortium of centers around the country, showing that we can do biopsy safely. It's a precision medicine approach. And if we biopsy these patients, we can get the genetic sequencing information that we can then tailor a particular treatment to each child. This is another study we've looked at using spinal fluid from patients to give better diagnoses rather than doing more invasive biopsies. So, these are small steps in the path. We have by no means found all the answers. This is a long road. I think we are not there yet for this tumor. Patients still get radiation for treatment, but we are looking at other treatments. Some are with chemotherapy. There are other potential treatments where we do something called convection enhanced delivery, where we insert catheters into the brainstem and then infuse different treatments, whether it's antibodies or other types of treatments into the brain tumor in the brainstem to directly affect treatment. So I think that I guess what I'm trying to say with this tumor is that without this innovation, we wouldn't have even been able to make these steps. And now just within the last 5 to 10 years, we've learned a lot more about it, but we have so much farther to go before we can really affect the treatment and find a cure for these children. So with that, I think that, but I think we're on the right track, and innovation in medicine, innovation in pediatric brain tumor, innovation in pediatric surgery in general is key to finding cures for diseases like this. So with that, I want to thank you, and I'm going to turn this back over to Terry.
Well, thank you, Suresh. That's fascinating to see that, and the technology's made a huge difference. We'll take a lot of questions at the end, and I've been trying to look at some of the things in the chat. But let me just ask you something, which is, as you look at the current technology, it's obviously a lot better than what we had 20 years ago. But what do you need? Like, what's if we could build you anything, right? If many of the people listening to this have the ability to build almost anything, what would it look like?
Yeah, it depends on the type of tumor. In pediatric brain tumors, you have the types that they grow and push brain aside. With that type of tumor, we can go in with surgery and physically resect it. And in those situations, we've been looking at ways of actually painting the tumor, injecting dyes that can actually, because sometimes in surgery, what can happen is parts of the tumor are very easy to, the key to pediatric brain tumor surgery is distinguishing tumor from normal brain. And sometimes it's obvious, but other times the tumor, because it comes from normal brain, can kind of blend into normal brain, and finding those areas can be challenging. So some things we're working on and other groups are working on ways of painting the tumor. Part of the challenge there is that finding companies that will do studies of their drug in kids can sometimes be challenging for various reasons. We are finding some drugs, like for example, there is a drug called 5-ALA, and it's used in adult brain tumors, but it hasn't really been used much in pediatric brain tumors. But now there are other companies. There's a study going on looking at a particular scorpion toxin that has been found to bind brain tumors. And we can use that to physically paint the tumor. So in surgery, you can actually see it fluoresce a different color, and then you know if that's tumor versus normal brain. So that's one example. Other things are where you have tumors like a DIPG that just subsumes normal brain. How can we selectively kill those tumor cells without affecting normal brain? And that's the challenge. And there are different technologies that we're working on, that different groups are working on, and that is really the challenge.
Great. Well, thank you very much. So it's also my pleasure to have Michael Muhonen here, who is going to talk about a different area of brain surgery and the technology related to that and the needed innovations. Thanks, Mike.
Thank you, Terry. So I came to Orange County in 1995, and one of the issues that haunted me then still haunts me now, and is how do we close the gap between the need and the availability of devices to children? And one of the first patients I saw when I got here, a little girl came in, she was about 13, 14 years old, severe spasms in her legs, and she had never really run her entire life. And I dug up this video to show you, what is presented to us on a daily basis. She comes in and she's taken Valium and all the other pills, but we had to do something innovative. She could no longer walk without full assistance. And can you see the video? So this is a girl, so you can see her, she has a 2-person assistance walk. She walks on her toes. Her toes are bent inward. And this is what I was presented with. Now, so my dilemma at this point, the FDA hasn't approved anything for children at this point other than maybe a pill of baclofen or some other muscle relaxants. So I had toyed when I was at University of Iowa with putting baclofen, which is a relaxant drug, right into the spinal column. So I called the IRB and said, hey, we have a child here. I'd really like to try something new that's never really been tried in children. And so Sarah became patient number 1, where we took a Medtronic infusion pump and put a drug in that called baclofen. And so the innovation was really trying to get this to the IRB. They had never heard of it. I had only done one in an adult a year before, and here she becomes the first patient in Orange County. Well, here she is 6 months with her pump. She's walking alone for the first time in her life. And in the next step, you're going to see she actually runs for the first time in her life. And this is a girl that had been going to therapy her entire life for her cerebral palsy. She was born prematurely, and here she is running with her gait pattern there. And so this is the problem we have. Putting this in an adult wouldn't have been any problem, but we had to see, all right, let's try something new for a patient like this that no one in the hospital had heard of. I had barely heard of it. And this is what we have a challenge with. What can we do to make these devices available to children? And this actually goes back a long ways if you look at the years. And I just wanted to share over the course of my work some anecdotal things that I have seen over the years, how this has developed, and things that I have helped to develop. And this is something quite simple. Can you see the slide?
Yes.
So this is a child that, this was out of a National Enquirer. Help, my baby has a flat head. Tiny helmets can shape an infant's head, but are parents going too far in their quest for babies? So this was invented and actually patented by a company who then put it on the New York Stock Exchange, made millions of dollars off something that some guy basically just took a piece of silicone and molded it around a baby's head. And whenever you go to a mall, you're going to see this all the time, something like this. And I looked at this and I thought, well, that's really not innovational. If you look back in the 1800s, when Lewis and Clark were going west, they ran into the Clackamas people. Look at their innovation. They wanted the flat of your head in the front and the back. You went to the higher level of heaven, basically. So look what they did. They just innovated and put the head on a board, put a board in the front. And William Clark's diary states that at the very bottom, with some degree of horror, he looked at this and noted they wanted the child's head to be flat and shaped to achieve the highest degree, essentially in death in the afterlife. Whoever had the most flat and pointed head would go to the highest degree. So 1806, we had innovation in pediatric neurosurgery before these helmets even came about. Well, one step further, as my career evolved, I became interested in dynamics of cerebral spinal fluid and pressure in the brain. About 2/3 of what I do involves excessive water in the brain, excessive pressure or cysts in the brain. And over the years, if we wanted to ever know what brain pressure was, we had to drill a hole in the head, put a sensor in there, like you can see on this dummy here, and then that ran to a cable that was on the bedside. Well, if you could imagine putting this in a child, you have to sedate the child, otherwise they're going to rip it out. If you have a child that's in an automobile accident, you could leave that in for a week, maybe 2 weeks, before you start to get an infection on it. And it really became a burden. And this is what got me interested in how could we do wireless technology? Well, people have tried many different techniques to try to, how could we measure brain pressure without a wire coming out of your head? So I spent the last 5 years working on a device called the Aura sensor, and we were able to get FDA approval for a quite simple device that on top of it, where it's a coil, up here by inductive coupling, you could power it. So you didn't need to put a power source in the brain, but we could measure the pressure merely with the power source put on outside of the patient's body, something like this. So we now implant this in the brain, close the skin over it, and then you measure pressure. Well, when you talk about problems with making these available to pediatrics, I have done 0 of these in my hospital. It's not approved for pediatrics, it's approved for adults. I've done 12 of them in the adult hospital across the street because it was FDA approved for adults. And if you look at other things that are typically approved by the FDA, halothane, for example, a common anesthetic drug, it's approved for adults. We use it all the time, but you can use it with physician discretion. But most companies, not pointing fingers at anybody, get devices approved for adults because it's easier, there's more patient population, and there's more money to be made is probably the #1 reason. So approaching something like this, here I was head of neurosurgery for 25 years, and I had difficulty implanting this in pediatric patients because it was FDA approved for kids. And this is some of the challenge we have to overcome to close that gap by making things like this available for the pediatric population. Well, my primary area of interest, especially more recently, has been hydrocephalus. So hydro meaning water, cephalus meaning head, and it's a Greek word. This is a picture that's 100 years old if you look at it. And it's a classical picture of a baby with massive hydrocephalus, a big head. And if you tap on it, it sounds like a cracked pot, McEwen's cracked pot sign. And the children have that typical look. They're emaciated, the eyes look down, the sun set into the eyes. And innovation on this has been a long time coming. It wasn't until 1940s, late 1940s, that something finally happened, and it was early 50s, and it's a device called a shunt. And it's basically a silicone tube that runs from the head, the water pocket in the brain, and the tube then drains the fluid into the abdominal cavity, or you can drain it into the heart, you can drain it into the space outside the lungs. But you can see the problems if you have a plastic or silicone tube that runs from there. You can have problems anywhere along the way. We had to innovate on this. Well, how did a shunt come to be? What are the problems with it? Well, it was basically a locksmith working for a Yale lock company in Philadelphia, and his son was Casey Holter. Well, Mr. Holter, John Holter, knew his son was going to die. You can see the child has a large head, he had spina bifida, and he lived until he was 5 years, but he had multiple revisions of Mr. Holter's shunt. And he made these devices right in his kitchen. And he sat here and put these silicone tubes together, put ball and spring valves in. And it wasn't as simple as putting in a straight tube. You had to regulate the flow. If you drain all the fluid out of the brain, the cortex collapses, the bones collapse. And you had to have a valve regulating mechanism. And over the last 60 years, since neurosurgeons have become involved in putting shunts in, the technology is basically focused on the valve and making the valve better. We now have valves that you can read externally, you can change the pressures externally, but they still have tremendous, tremendous problems. This is a classical picture of a shunt. The child comes in with the shunt not working. If you look at it, the shunt's tied up in a knot, spontaneously tied up in a knot, harder than I could probably put tie in it, just by the child's movement. And you think, well, how could that tie up in a knot? You have a 4-foot-long tube or 3-foot-long tube in a body, and pretty much anything can happen. The tubes can migrate out. They can migrate out to the rectum. The valve up here is a device here that can plug up with debris. You can see blood in the tubing, and they're problematic. It's just not a system that the body was made to live with forever. And if you leave it in long enough, this is what happens. The body basically rejects it by encoding all the silicone tubing with calcium. And this is a shunt where I tried to put all the pieces together that I took out, and you could see they kept breaking because they had extensive calcification on all these pieces of shunt tubing. So this has led me to the innovation that I really am pursuing now, and I think will be the answer and the holy grail for many neurosurgeons, especially for pediatric neurosurgeons. Can we create a smart shunt? Can we take the smart people at ULP, the smart people at Medtronic, the smart people in the industry, the engineers, can we create a smart shunt? In other words, can we make a shunt that goes in the brain that can respond to pressure gradients. So when you stand up, can we stop it from over draining? Can we put something on the outside of the head so we can program it to drain more or to drain less? Can we measure the pressure through the shunt tubing as the pressure comes in it and the pressure in the brain? How can we disocclude the piece of the catheter that's in the brain? That's the number one reason these things quit working, is because the catheter in the brain plugs up with debris. How can we disocclude the catheter that's in the heart or in the peritoneal cavity? How can we disocclude that without having to do surgery? And the problem is the average patient that has a shunt has about 10.4 surgeries in their life. And so if you come in, I can tell a mother, if you come in with a newborn baby with a shunt, there's a very high chance you're going to have a dozen surgeries on this, canyon infections and revisions. And this has led us to where we are today, trying to make this better. How can we create a smart shunt with what we have? And I go back to the reason we're here, and that's innovation. How can we create something innovational that's going to cure the problem? And that's where we're at now. And how do we overcome the inability to close this gap? How can we get these devices that we do develop into the children's hospitals? And, when I talk about, stop sharing here for a second. When we talk about innovation, I go back to that first patient, Sarah, that I showed you that had the baclofen pump. As the pump evolved, it got used more and more frequently for pain. So adults have chronic back pain and things like this. And as that evolved, somebody came up with the idea of trying a canotoxin in this pump. And it's a ziconotide is the name of the drug, and it's dissolved from a cone snail. Well, the cone snail derivative, it's the conus magnus, is what you're essentially putting in there. It's 1,000 times stronger than morphine. So we can now put a derivative of a snail shell, the cone snail, directly into the intrathecal space, and the patients don't get the narcotic addiction with tremendous relief of pain. And I have probably 20 patients now that I have basically put snail juice into their intrathecal space, and it works very well. And that was all because of innovation. Is it approved for children? No. When I put it in the hospital, do I have to get approval from the IRB? Yes, every single time. If I do it outpatient in my office, I can do it. But these are the issues that we have to overcome. So I hope I've piqued your interest in this a little bit, because there's somebody out there that's going to help us discover a smart shunt, a shunt that will obviate the need for 10 surgeries in the future. And you'll stumble upon things like a cone snail someday that will make this innovational and a cure for many children.
Well, thank you very much, Michael. It's wonderful to hear this. And between snails and scorpions, I think we're we're making good use of the biological environment. I heard you both address certain topics about that some of the issues that are impediments to pediatric technology development. One of which is that we're really borrowing so much from the adult world rather than starting in the child world. But one of the things that I often hear phrased as one of the reasons you do that is because there's a sort of an ethics question, which is, shouldn't we be trying this stuff in adults first and making sure that it's okay in adults before we do it in children, even if children are the primary target? What do you both think about that? Are there situations in which it's okay to try something in a child first if they're the one that's most likely to benefit?
I do. If you look at even every publication for marketing that comes out of CHOC, it will say something like, we want to treat our patients because they are the most vulnerable in society. Okay, so they are the most vulnerable, but they are also probably the most needy. Fixing a problem when a child is now 18 rather than fixing it when they're 1 or 2 years old, you've lost that window of development. And so, yes, I agree, they are the most vulnerable, but I would say they are the most worthy in the population that we should seek after.
Yeah.
Suresh, what do you got?
Yeah, no, I think it's difficult. For in the DIPG example, we went many years without really making any advances in even understanding the tumor because of the ethics of, okay, do we do a biopsy in a child? You can't just do the biopsy to if you're going to do a surgical procedure on a child, it needs to have some direct benefit on that child. You're not going to do it just to learn about the tumor that might affect kids in the future. And that makes complete sense. So when we offer a surgery, you have to do something that's going to benefit that child potentially. Now, in addition, if then that tissue can be studied and we learn more about the tumor that we can then design better treatments in the future, that's a plus. So, for many years with DIPGs, it was thought to be the wrong thing to do any biopsies because the side effect profile was too high. We didn't have anything to offer them. And that's changed now because we can do it safer and we do have something that we can offer in a particular biopsy. And as we do that, we can offer something and then learn more about the tumor. And some of these tumors are not, this particular type of tumor is really more specifically in kids. It's not really seen much in adults. So I think in the past, people thought of these tumors as GBMs of the brainstem, and GBM is a particular type of high-grade tumor in adults. And now we're learning that they really aren't. They're a distinct type of tumor and they're very different. So I guess in answer to your question, there are situations where certain things cannot be tested in adults first. And if we don't learn more about the tumor in kids, we cannot make any advances and these children continue to suffer. So I don't think there are easy answers to this, but we do need to be able to understand these situations better.
Well, and thank you both for those answers because I strongly agree with that. I mean, I think that there's a tremendous fear in the device and pharmaceutical industry of approaching certain childhood diseases because of this sense. But I think partly because of that fear, we may end up depriving children of things that they need. There may be things that don't work in adults that do work in children and would be discarded if they were attempted in an adult first. One of the other barriers that comes up a lot is the potential, the concept of reimbursement in the pediatric world. And a question came in about the relevance of things of incentive programs like the breakthrough device designation and whether that can help device producers to focus more on the pediatric market. What do you think? What types? Let me expand on that question a little bit. Which is what types of incentives do you think would be helpful to really get more, not just basic research, but take some of these basic questions and turn them into distributable, marketable devices and technologies that could really affect children, given that the pediatric market size is always going to be smaller than the adult market size? I mean, what kind of regulatory changes or what do we have to do to get these ideas out there in a commercial environment?
Mike, you want to go ahead? You can.
Yeah. Well, the issue is difficult, and because when you get a consent for a device, that some of the things I've talked about, you have to get an assent from the child also. And it's always bothered me a little bit when I'm telling an 8-year-old child, yeah, I need your permission to put in this medical device, this pump, for example, because it's not approved. And we probably have the best IRB in the world as far as helping us try to get things through. But so there's an ethical dilemma that we have to overcome here. And I think our approach, instead of approaching them as the most vulnerable, I would approach them as the most needy. And I would. Industry is going to have to contribute to a regard that they're helping the betterment of the cause, and there's not as much money to be made in hardly any of these childhood diseases. If you look at Spinraza, for example, it's a drug we give for spinal muscular atrophy. There are so many children with that disorder that the drug is about $1 million a year. They get one dose of it every 4 months, and you put it right into the spinal fluid space. The adults that live with it was where the trials were done on the bulk of this. And the drug companies had far easier time getting these done with adults. And the other limitation in children is you're limited by size on many devices. You have a baby, you can't put a pump in a baby. You can't put some of these stereotactic frames that Dr. Magge showed you on a baby. So this is going to take industry involvement in this to make this happen. We can make the greatest inventions, but we have to make them accessible for, you know, a child that's a foot long.
Yeah, I think it's a hard question. I think I'll go back to an example that we talked about, the tumor paint. A few years ago, I remember trying to bring some of the tumor painting technology to kids. So there's a drug that has been used in adult brain tumors where basically you inject the child before surgery so that in surgery the tumor can glow, and it's easier to distinguish tumor from normal brain. And it's several years ago, there's a lot of traction for this drug in adults, and I was wondering, why aren't we doing this in kids? And as I tried to approach the company to see if we can do this in kids, I think there was a hesitancy for A, it's a smaller market, and B, the difficulty of getting FDA approval in children and safety studies. And I understand that. I mean, I think there are, of course, valid reasons why some of the safety barriers are there for kids. But it's something that we need to deal with as well to be able to get these types of drugs to kids.
Okay. I think it is, you know, the FDA is definitely interested in finding ways to do this, and they created a program specifically, several programs specifically to look at this, and there are some incentive programs to try to bring pediatric technologies, and actually this has been more applied in the drug world, but in the technology world there's definitely room to do this in terms of innovation, but it's difficult. And I think one of the things that you both have shown by the history here is this sort of the tinkering, you know, the sort of intelligent tinkering by the clinicians at the front line who just solve problems, or you know, the guy who was sort of inventing shunts on his kitchen table, right? Just because it's the only way you can do that. And I think finding pathways that can take that kind of insight and that kind of tinkering and transform it into meaningful products for children is tough. I mean, most things that you try aren't going to work. And so there's that you have to try these in a safe environment. In other words, when you're using these technologies from adults and children, maybe it's the first time, I mean, Mike, you're doing something, creating the first back open pump for children. You knew a lot about what the safety issue would be here. You don't have to try this on 100 children and go through multiple iterations. You can get a tube into the cerebrospinal fluid, and you have pumps that exist, and you can put these things together, and you know what the drug does. But it required that knowledge, and so I think I'm trying to get at this idea of how do we encourage, how do we teach our young neurosurgeons and other clinical specialties to sort of be the safe tinkerers, the people who, without putting the children at risk, can ask and try these things in a safe and ethical way that still pushes things forward, sort of one patient at a time.
I love that question because I view myself as a safe tinkerer. And 4 years ago, one of the orthopedic surgeons came by, and he had a child with a completely fused spine. He said, "Hey, I need you to get a catheter in there that's going to last, and that we can squirt this drug in her spinal fluid continuously." He said, "But the FDA has it only approved to put it in the lumbar spine." But logically, the target for his drug, via Spinraza, was the motor neurons up in the cord. Well, there's no spinal cord in the lumbar spine. It's all roots. So we took a device approved for the lumbar spine, a Medtronic little lumbar shunt that we would normally use. We put an angiodynamics port attached to that. We put some other interstices. So basically I put it together in my garage one night, trying different pieces, put them together. And I put it in the first child, and this thing worked like a charm. And still to this day, 3 years later, we still have the same port in this girl right here on the base of the skull. And it was only by trying that, it was a lot of work to put a catheter in the neck for risk of the spinal cord. And, it comes big circle back to fruition. The president of the group looking at this drug puts a slide up at his talk last week and said, we've just discovered that the drug works best where you inject it. So it works best if you put it in the neck, if we can get the catheters up there, because it's right on the target. And so logically, it's where the drug has to traverse the least amount of distance. So a safe tinker is, first, do no harm. And second thing is you have to be a Boy Scout. You have to leave the area better off than how you found it. And do no harm, leave it better than how you found it has to be how we approach these patients as we do safe tinkering.
I think there's this sense of, it's a different kind of research. We're very used to this idea of statistical research, where you try something in 100 or 1,000 patients and see what happens. But I think there is this, particularly when you're dealing with surgical procedures, there's a real sort of N of 1 kind of research. And that's made possible because you have very big effect sizes. I mean, you take someone who had never walked before and now she's running, right? You take someone who's going to die of their brain tumor and now they're not. I mean, these are very, very big effect sizes. A quotation, I believe, from Ramachandran that I'm very fond of is to say, if you have a talking pig, you only need one. The point is that you have a large enough effect size, you can prove success with a single patient. But it's hard, right? I mean, it's hard because you're a tinkerer, but you can't try it over and over and over again. You've got one child, one shot at it. So so again, I would come back to this idea of like, how do we encourage the next generation to do this? Because we all grew up in a regulatory environment that was still highly regulated, but it's been moving toward more and more regulation. Things have been moving much more toward closer supervision, towards more patient protection and more oversight. And I think some people end up finding that very scary and potentially daunting. How within a regulated and closely monitored environment do we encourage our junior members to take these kind of risks and to use their insights to make a difference for their children?
Yeah, I think, that's a tough question. It's a balancing act. Like Mike said, is our first motto is do no harm. And, I always tell our residents, do the right thing. In the end, if this were your child, how would you treat them? You treat them as if they were your own child. So, but that being said, doing the right thing is also asking questions and questioning what we do. How can we make this better? After every surgery, I talk to a resident, say, okay, it could have been a very routine surgery, but you can always learn from everything you do. There's never a routine procedure. So at the end of the surgery, you critique it and think about, okay, how did we do it? Is there something we could have done better? Should we have done this a little bit differently? Could we have made the incision bigger or smaller? And you're constantly learning. And so that's what I always encourage our trainees, fellows, residents, to always question what could we do better? So from whether it's a surgical technique aspect, or, are there certain devices that we can use to make the surgery less invasive, less painful? As we talk about precision medicine, that's, people talk about that is, smaller incisions. And And you're right. I mean, there are good reasons for the regulations that we have because, of course, history has been filled with instances of poor research and poor treatment of people and inequity. And so I think there's a good reason for the regulations that we have. That being said, that doesn't mean that we cannot innovate, and there are mechanisms that we can use to do that, and to figure out how do we do this better within the framework, but do it safely. And so, yeah, I think these are the questions that we always think about on a daily basis.
Well, I love the way you put it. I mean, the idea is that as innovators, you should never be satisfied, right? There's always something you can do better, and particularly in medicine. I mean, we're going to look back 50 years from now on what we do today and think, oh my God, if only we'd had the thing, that we now have, right? Because how did they possibly do this without the thing? Right.
Well, that's what, we talk about that on we're doing surgery, and there's some common instruments that we use. Like, for example, we use electrocautery, and it's a device, it's an electrical device that you basically, you can coagulate blood vessels. So it is a super common thing that we use in surgery. It's how we can open up incisions without creating a ton of blood loss. But you think back 100 years and people did surgery, and we think, how the heck did they do surgery without this? Or even MRIs. I mean, think about MRIs. MRIs are really within the last 30 years, right? In the 90s and beyond. Maybe in the 80s they were innovated. But people were still doing surgery in the 70s and 60s. And how are they doing that? And we think now it is such standard of care. And we couldn't imagine doing complicated brain surgery without an MRI and knowing exactly where we need to be. So we think back, my gosh, 50 years ago, they didn't have this. And so we always think, okay, in 50 years, like you said, what are we going to think back? They're going to think, what were those people doing 50 years ago? They were opening up skulls to do brain tumor surgery. I can't believe, will that be what people will be talking about? In the future,
saying, why did we ever even let humans do this, right?
Exactly. I can't believe humans were actually doing this. They didn't have robots back then. There will be things that they will talk about, and I can't believe in 2020 they did, 2021 they did it that way. So
there's a couple questions in the Q&A. There's one to Mike talking about your smart shunt design. I don't know if you have it in front of you, but I'll read it.
Yeah.
It says, in your smart shunt design, can you develop bioimplantable or biodegradable devices, and possibly looking at using calcium pump technology or other kind of bioengineering? And these sound like, in some cases, microfluidics questions.
Yeah, that's from Calvin. Thank you, Calvin, because I love your thinking. And I love the fact we have about 30 bioengineers out there, and I got their minds thinking, because this is what you want to do, So basically saying, can you just pump the water out of the brain, basically? Or could you put a tube in there that degrades, or a tube that's made out of your own cells? And that's exactly what we're thinking as we progress with a smart valve or a smart shunt. Can you put a liner made out of your own cells that dumps the fluid from the brain back into the venous system, so you don't have to have a 4-foot-long piece of silicone with a valve in there? So that is a good direction of bioengineering. Engineer your own cells to make your own internal shunt. I like the idea of pumping the water out, but I envision it like when I lived in Iowa, we had these sump pumps where your basement would flood, but the sump pump would pump all the water out of the house. You need a sump pump of the brain, basically, that would have to be tiny to get the spinal fluid out of the brain. But this is the direction you have to go. We're chopping the top of the weed off every time by just putting a shunt in there. You still have the basic problem. You still have a failure to absorb the spinal fluid, and we're not solving the genesis of the problem.
There was another question, which was, how well do insurance companies respond to reimbursement requests for these kind of things? And I don't know what experience you guys have both had with getting approval for the use of these technologies in children when they are being used, let's say, very, very far off label.
Well, they hate it. They don't like it. I remember we did a vagal nerve stimulator in a child after these came out, and they said, we will not pay for it. These were $12,000 implants. And finally, they broke because it had there was merit to putting it in. But these are all expensive devices, and they don't respond well to requests for new therapies. They'll just go by what their paperwork says. These are not approved.
Yeah, I'll give an example from a different topic that I didn't really talk about today. It's a topic called craniosynostosis. Craniosynostosis is a condition in babies where part of the skull fuses too early and causes a misshapen skull. And this misshapen skull can, in some instances, lead to increased pressure on the brain. The traditional surgery for that is a big open surgery where you open up the whole scalp and take the whole skull apart and put it back together. And that's the traditional surgery. It still works very well, and we still do that. In the last 15 years or so, 20 years, we've been doing more minimally invasive surgeries where instead of doing that, we can use a camera and just take out the part of the skull that's fused. But we do it when they're really young. And then after surgery, we put them in a molding helmet. And that molding helmet helps to reshape the skull. Before coming here, I used to be in Washington, D.C. at Children's National Hospital, and I started our endoscopic craniosynostosis surgery program. That was, I don't know, 12 years ago or something like that, 15 years ago. And at the time, it wasn't being done very much there. And so I still remember the insurance companies, the success of the surgery was dependent on these babies wearing a helmet after surgery. And I remember a lot of insurance companies would not pay for it, despite the fact that in their, the surgery is much less invasive. The kids spend much less time in the hospital, less blood loss, and it costs less for the insurance companies. But they didn't understand this. So I remember it took a lot of phone calls with insurance companies so that they could understand, talk to their medical directors, understand what this is, why the helmet's for. And eventually, as time went on, more and more the insurance companies understood this and they started paying for it. So I think it's just a process and educating them as to what we're doing.
I have to say, my experience with this has been sort of mixed, which is that I was once talking to the medical director of an insurance company and trying to get approval to do deep brain stimulation off-label in a child. And the question that this man asked me is, he said, how many of these do you anticipate doing per year? And I was like, oh, 5 or 10, maybe. He's like, then I really don't care, because no matter what the thing costs, it's not even going to show up as on our budget. It's too small because it's rare. And I think that aspect of it, combined with the fact, and this was a comment that was made earlier, which is that you have to look at the lifetime medical cost of not doing these procedures. And I think you say, look, this is rare, so the financial risk is low, and the lifetime cost of not doing it is quite high. These children sometimes will die if you don't do these things, but sometimes they're not. They're left with significant disabilities, but nevertheless lead very long lives that become very medically expensive for the insurance companies. And so I think being armed with that information when you get these medical directors on the line or when you're filing the appeals papers actually does, in my experience, it's often been possible to do this. But again, it varies a lot. I don't think there's much uniformity to it. I do want to sort of bring this to a close. I think we've had a very fascinating discussion here. But I would like to just give you both a chance to just just talk about like where what do we need? How do we create innovation? We have an audience here who's listening to us. They're coming from all different places, from the technology world, from the business world, the investment world, the educational world, the medical world. We have people who are all of the things that are brought together by BIAL, by university lab partners, by the various innovation groups that have invited us to this talk, and that are really trying to bring people together. And I think trying to think about all of those people listening to us. We have a tremendous program in innovation in nursing, for instance, at CHOC. And how do we make a difference? What do we need to really drive innovation so that whatever we're doing now, we can double it or triple it or quadruple it in the next few years? How do we encourage that?
Well, you need game-changing ideas, game changers that make children's life better, and devices that lower the burden to the healthcare system. 1 shunt versus 10 shunts in your lifetime, for example. And you're going to get these from maybe even someone in this audience that's listening that has a light just went off in their brain. Hey, why don't we do this? Why don't we take this device and we'll put it in the patient's bladder, and that will stop all urinary tract. Something, it's just going to be something will trigger. And I would urge everyone with these ideas, collaborate and talk to people about it. And specifically, if it's something that a physician can get involved in, approach them. And I've had nurses come up with wonderful ideas at CHOC and said, hey, why don't we try this? And things that have just worked beautifully for things that I would have never thought about, but they're with the patient all day. So, when you have these ideas, don't let them smolder, get them out there and collaborate with others. And I think that's one beauty of ULP. You have so many people in one big setting that you can do that. And collaboration and putting the ideas out there and trying different things is how we're going to make progress in this.
Yeah, I think that what we're doing today, and this, even with what bringing together people from all different backgrounds, clinicians, scientists, engineers, business people, everybody. I think the key is collaboration, bringing people with different backgrounds, different ideas, different perspectives, because we can't just have one perspective on, we need to bring in ideas from all different backgrounds, and that's the only way to tackle these difficult problems. Like what Dr. Muhonen said is, we have from everybody, nurses, PAs, docs, everyone in the healthcare system, but engineering, science, bringing everybody together in a forum like this and collaborating, because that's really the only way to do it. What Mike is doing with his smart shunt technology is amazing. And I think, in pediatric neurosurgery, CSF problems, hydrocephalus is one of the biggest issues we face, and has such quality of life implications for these children. So what Mike is doing is amazing, figuring out a better way to do it, because this is an issue that's plagued hydrocephalus has been such a problem for kids for years, and you can have multiple shunt problems. And so, like what Mike is doing, finding a better shunt, or finding a better way to take out brain tumors, or a better way to treat skull shape anomalies. It takes a panel like this, bringing people together to bring out the best ideas.
I completely agree, and hopefully many of the people that have just been brought together now by OC Life and by this meeting will be contributing to that. Thank you both for your comments and for sharing this fascinating view of neurosurgery, these neurosurgical issues. As we talked about before this, we're all very used to this, but I think many people in the outside world are not quite used to seeing the technology as well as the gory details of this, and recognizing the tremendous skills and advances that have already been made. So if we can continue this, the kinds of problems that we can solve and the way we can help these children will really be tremendous. So thank you both. And with that, maybe I'll bring this to a close.
Thank you.
Thank you, everybody. And our emails will be available. If you guys have questions, just shoot us some emails. Anything you think of, these bright ideas, let us know.
Absolutely. Thank you, everybody.
Bye.